I'm the CEO of We're going to be recording tonight's session because some people can't actually join it. And also just to say that we will take questions at the end, but when the speakers are speaking, if you do want to put questions in the chat, if you're worried that you won't remember them at the end, then please feel free to do so, and we'll get to them at the end. So I'm delighted to welcome you to our latest talk in this very exciting series, diving into our gene pool. And this talk's entitled Three-Parent Babies, We Can, But Should We? And just a little bit of background about genetics. We exist as an organisation to raise awareness, educate and test the community for Jewish genetic disorders. JGD is a term that we often use to describe genetic conditions that are more common in people of Jewish ancestry, and that includes Ashkenazi, Sephardi and Mizrahi backgrounds. Currently, genetics test for 47 of the most recessive Jewish genetic disorders, all of which are life-limiting and have no known cure. So we started screening back in 2014, and since that time, we've screened almost 7,000 young adults, educated tens of thousands, and identified numerous carrier couples who we have helped go on to have healthy families. You may have heard that we have recently been appointed by NHS England as the official engagement partner for the new NHS England, for the new Jewish BRCA testing programme. And we're working very closely with High Cancer Care on the delivery of this programme. Since the programme launched officially at the end of January this year, over 21,000 people have actually signed up to be tested through this first of its kind groundbreaking programme. So BRCA gene faults are 6 times more common in those with Jewish ancestry. That's actually the Ashkenazi figure, and I think it's 3 times more common in Sephardi compared to the general population, thus emphasising the importance of the programme for our community. So it really is fantastic news that this programme has been launched by NHS England. So both the recessive screening programme and the new Jewish BRCA testing programme give people the knowledge and the power to make the best decisions for theirs and their future family's health. For more information about either of these programmes, you can go onto the genetics website, which is genetics.org, or email the genetics team. We are privileged to welcome this evening our esteemed guest speakers, Professor Joanne Poulton, Dr. Doron Behar, and Rabbi Gideon Weitzman. So at this point, I'm going to pass over to Gideon Schwartz, our Outreach and Education Executive, who will be chairing this evening. Over to you, Gideon.
Thank you, Nicole. So when we think of DNA, we mostly think of the DNA inside of our nucleuses. And that is the DNA that really we at Genetics are sequencing, and that's what we look at in both our recessive and BRCA testing. But in addition, there's a small amount of DNA actually inside your mitochondria, 37 genes to be precise. And many of that actually makes the enzymes which we use in respiration. So we inherit our mitochondria, and thus all the mitochondrial DNA, directly from our mothers. Nothing comes from our fathers. And these genes can sometimes have mutations in the same manner that the regular nuclear DNA does. And when they do, a mother will pass them on to her children. At Genetics, we firmly believe knowledge is power. And the disease that we screen for via our recessive screening panel can all be prevented using existing reproductive technology. Similar technologies are emerging to prevent these mitochondrial disorders. However, is fraught with potential technical and ethical issues. So I'd now like to introduce our first speaker, Professor Poulton. So Professor Jo Poulton is Professor and Honorary Consultant in Mitochondrial Genetics at the University of Oxford, where she worked on mitochondrial diseases. Her data on the transmission of mitochondrial DNA were a breakthrough for the feasibility of mitochondrial prenatal diagnosis and ultimately for the high-profile mitochondrial replacement therapy of 3-parent babies. And she suggests that drugs may be used to harness the natural cellular quality control systems to alter the balance favorably and reduce disease severity. But before her, I'd like to introduce our first speaker, which is Dr. Doron Behar. So Dr. Doron Behar has been a pioneer of the genomic industry for more than 20 years. He's a doctor in internal medicine, critical care medicine, and medical genetics, and he holds a PhD in molecular genetics and population genetics. As the founder and CEO of Igentify, Dr. Behar has overseen the development of innovative suite of digital tools designed to scale the provision of genomic medical services to more people worldwide. Igentify software helps clinics improve patient enrollment and consent processes, provides labs with faster and more robust interpretation of genetic testing results, and enables genetic counselors to serve more patients with enhanced focus on patients at risk. Prior to founding Igentify, Dr. Behar has served as Chief Scientific Officer of Family Tree DNA and Chief Medical Officer of Gene by Gene in Houston, Texas, a genomic research center and one of the largest genetic testing labs in the United States. So, Dr. Behar, over to you.
Thank you very much, all. Am I well heard? So let me just I need to. You need to allow me to do the screen sharing, Gideon.
Yeah, 1 second.
It's a pleasure to be here. As the Gideon,
you should be able to share now.
Let's do it. So, can you see my screen now? Perfect. So it's a pleasure to be here, and it's a pleasure to let you know about the mitochondrial DNA and the evolutionary aspects that relates to the Jewish people. That was a part of a series of studies and paper I conducted already, I think, 15 years ago. And it's going to be a completely different view of the mitochondrial DNA compared to what we are going to hear later on with respect to diseases. So please feel free to later on ask questions regarding other components that we might not discuss here and so forth. So first, we're going to make a little review of genetics, which is going to be quite focused on what I am for. So it's probably known already that each human cell has 46 chromosomes. 22 of them are pairs of autosomes, and one pair is the sex chromosomes. So you can see it here, 1 to 22, and then X and Y in this case, which will represent a male. Other than this, we have the mitochondrial DNA, which should not be confused with X chromosome. It is a different small molecule, 16,000 nucleotides or bases in its length, and it is actually part of it's not integrated yet into the autosome or to the genomes. And it is very important to notice about here that there are some things that I will try to teach you about the chromosome and the sex determination. I'm going to make it very visible on the Y chromosome, and we can immediately understand the mitochondrial DNA afterwards. So basically, as a geneticist, we were trying to look for molecules that might allow us to check the paternity or the maternal lineage, the paternal or maternal lineage of our ancestors. And for this process, I want to show you this, which you will all immediately agree and accept. So, you know, mothers have 2 X's, father have X and Y, and then each of them must give one of the 2 chromosomes that they have to one of their offspring or kids. And then you can see here something very, very clear, that the Y chromosome can only come from the father, which means that this one will mark a paternal lineage, because then this father will get his Y chromosome for his father, and so forth and so forth and so forth. And basically it means to us that if 2 males are sharing the Y chromosome, we actually can calculate when was their most recent common ancestor. And we have to put it in the context of things that many West Eurasian populations are doing, which we are actually also transmitting the surname from father to son, and so forth. So we have now this great tool called the Y chromosome that can teach us about whether people that are carrying today the same surname might have the same common ancestors, or it is just by a coincidence. For example, they both had the same profession and they chose to call themselves following this profession. So that has been quite widely used worldwide. You probably know of the major companies that are serving the genealogical audience. And taking this into your mind, please let's focus on the mitochondrial DNA, which is the basic theme of this particular talk. So if we are looking at the parents, so these are great grandparents, and they have their autosomes, which are marked in this bigger chromosomes appearance. We have the yellow one, which is the Y chromosome, passing from father to son to son. And then we have the mitochondrial DNA, which is passing from mother to all of her children, but only the mothers, the future mother, will further transmit it. So basically, if you are a male, half of your autosomes will come from each parent, your Y chromosome will come from your dad, your X from your mom, and your empty DNA will come from your mom. If you're a female, then again, half of your autosomes come from each parent. You have X from both dad and mom, and your empty DNA come from mom. So a male can trace the paternal ancestry, father of father of father of father, and mother of mother of mother of mother using the mitochondrial DNA, and all the other ancestors should be checked differently. We are not going to enter this today. If it's a woman, she can trace the mother of mother of mother, because as we said, women don't have the Y chromosome. And I'm going to turn you all into population geneticists quite fast. So let's say that there are mutations or variants that are basically here shown as color. So there are some mitochondrial DNA that have a mutation, a color that differs them from one world to another, from one place in the world to another place in the world. As you can see, we have here Europe. There is no color, but as you can see outside of Europe, there is a lot of different colors. Just an example, but naturally, if I'm going to show you now an individual which you are not familiar with, and I'm going to tell you it doesn't have the color, is he European? You would likely say that he is not European because we didn't see any color before this among Europeans. So that's very basically what we say, but we have many more than one mutations, and we should look to mutations at a very interesting way that they evolve. Because if we are going to read this well-known phrase from Small Gods, it says that events just happen one after another. They don't care who knows. But history is different. History has to be observed. Otherwise, it's not history, it's just, well, events happening one after another. If I'm going to switch this to evolution and mutations, then you are going to get this new piece, and it was modified without permission, I admit, but mutation just happened one after another. They don't care who knows, but evolution is different. Evolution has to be observed. Otherwise, it's not evolution. It's just, well, mutations happening one after another. And the concept is very, very simple eventually. Try to think that there is a root. So there is a first edition of a book. It was published. It was well used for a class of biology. And then in the next year, we need to create another book. And people were copying the previous book, but there was only one mistake in one letter out of the 1,000 pages that are in the book. Now we are going to have two versions of the book in the class, in the world. Another year passed, and we are now creating another book, and there is another mutation because people copied it. And this is how we can see that we are starting to have various editions, and naturally, everything, this version of the book must have the mutation of this book, which is going to the beginning. And that's how it really looks. If we are going to look to the mitochondrial DNA and Y chromosome of the world, we can see that there are many, many different colors in the world, but I want you to be very careful when you are looking at something. As you can see, there's a lot of colors here, but as we move to America, we see that the colors are becoming less and less in numbers until in South America, there's only one color. And mimicking this is the mitochondrial DNA that is detected in populations. And you can see that as we go south on America, we can see that there are less and less colors. And we said that evolution has to be observed. So now we have to ask ourselves, where do we have this color? Can it tell us anything about migration? And we can say, did people come to America from Australia, from mainland Asia, and so forth? And what that you can see here is that this color appears also in East Asia. And that is actually telling us that during the Ice Age, while the water were much lower and Beringia was open, there was a land bridge between Asia and North America, and this is how humans moved inside the Americas. We can say much more things than this. We can actually build the phylogeny of the human mitochondrial DNA. And we can actually say, we actually know now that roughly 200,000 years ago, we all shared one common maternal ancestor that split, that today we have branches that are coming only from two of her, so to say, children along the generations. And we can see something very interesting here. We can see here that in purple, we see the huge variation of Africa. In blue, we can see the variation of South Africa, primarily among the Khoi and San people. And actually what you see here is the out of Africa lineages. So it shows you immediately that the plethora of genomic diversity in Africa is much wider than what we see all over the world. And that is actually one of the pillars of the understanding of the out of Africa theory and the movement of humans. So we can say now that using the mitochondrial DNA, we can trace the movement of human throughout the world, from Africa, outside, branches going to the Fertile Crescent and to Europe, branches going to East Asia, and all are contributing eventually to also move into the America. The time frame that the human migration out of Africa is 65,000 years, more or less. And with this, we are going to switch to read together a short paragraph about evolution. So you are all convinced that is happening, as I said, and then we'll move to the Jewish genetics. So the European Commission has just announced an agreement whereby English will be the official language of the European Union rather than German, which was the other possibility. As part of the negotiations, the British government conceded that English spelling has some room for improvement and has accepted a 5-year phase-in plan that would become known as Euro-English. In the first year, S will replace the soft C. Certainly, this will make the civil servants jump with joy. The hard C will be dropped in favor of K. This should clear up confusion, and keyboards can have one less letter. There will be growing public enthusiasm in the second year when the troublesome PH will be replaced with F. This makes words like photograph 20% shorter. In the third year, public acceptance of the new spelling can be expected to reach the stage where more complicated changes are possible. Government will encourage the removal of double letters, which have always been a deterrent to accurate spelling. Also, all will agree that the horrible mess in the silent E in the language is disgraceful and it should go away. By the fourth year, people will be receptive to steps such as replacing TH with Z and W with V. During the fifth year, the unnecessary U can be dropped from words containing O, and after this fifth year, we will have a real sensible written style. There will be no more trouble or difficulties, and everyone will find it easy to understand each other. The dream of a united Europe will finally come true. And after the fifth year, we will all be speaking German like they wanted in the first place. That's how evolution progresses. Step by step, minor steps that eventually bring a completely different seeming situation. We're going to now switch into Jewish genetics, and we are going to look how we can utilize mitochondrial DNA in the study of the Jewish ancestry. So the historical records are suggesting that from an ancestral deme at the Levant, Jews moved both east to the Iraqi and Iranian landscape, and also to Europe to create the Ashkenazi Jewish population. Naturally, as geneticists, we are not trying to please other disciplines. We are trying to show what our data is showing. It might be in concordance or not with what historical records are telling us. But I'm not going to go into the complex moving of Jewish people, but I'm going to show you immediately that if we look to the mitochondrial DNA of Ashkenazi Jews, then we want to look to the colors that are in the regions that Ashkenazi Jews are considered to be part of, either the non-Jewish Mediterraneans, which their colors are here, or the non-Jewish Europeans, because people are saying they came from the Levant into Europe. And if we are going to take a look at the Ashkenazi Jewish pool, we see something very interesting. You see that it doesn't look like this. There's a lot of yellow here, a lot of yellow. And this yellow is only 6% in non-Jewish Mediterraneans, and it is in 7% in non-Jewish Europeans. But certainly among Ashkenazi Jews, it's a huge amount. We can also see that there is an H, which is similarly frequent in the other pools, but we see that N1 is very dominant among Ashkenazi Jews. We can't find it at all in European, and we can find it in a little bit here in 3% of the non-Jewish Mediterranean. So that's quite interesting if we are going to take a look of it. And that is actually where we are starting to understand that some demographic factors have shaped the ancestry of mitochondrial DNA of Ashkenazi Jews. So the questions now goes into us looking to this component, which is called K, and trying to resolve it deeply and trying to understand if we can see any relations among Ashkenazi Jews and between Ashkenazi Jews and their hosting populations. So, of course, your job as a PhD student is to convince your mentor that you need to travel in the world to collect samples, which is what I did. And as you can see here, we have collected samples from a lot of places around Europe, North Africa, and the Middle Eastern, and we have contrasted them with samples taken from people who self-identify as Jews, and we only looked into the component that I said, which is very large, the haplogroup K. And if one is going to look at this very complex figure, this is actually the phylogeny, what I showed you globally before. This is the phylogeny only for the K ancestry when you are looking to the entire mitochondrial DNA sequence. And you can see that there's a root here, there are mutations defining the root, and then there are splits, and then there are further splits and so forth. What I've done here, I've marked for you in blue where the Ashkenazi Jewish samples are all collapsing into 1, 2, 3 lineages. If we're going to take a better look at this, we are going to now to see by looking to the visualities of this. So in light blue is the Ashkenazi Jews. In dark blue, it's Sephardic Jews. In red is European. So you can see that we have here one lineage, which is separated by a few mutations from its closest similar mitochondrial DNA. And you see it is shared between Ashkenazi Jews and Sephardic Jews. We can see here also that there is Ashkenazi Jews and non-Ashkenazi Jews, and there is one individual that self-affiliates as European. We didn't know this was taken from collaborators of mine. We didn't know his religion, but we only know that the village he was sampled is called Hemlinsky. And then you can see that the same is happening here. So now we're in a situation where we know that there are 3 different Ashkenazi lineages dominated the Ashkenazi mitochondrial DNA K haplogroup. And these are virtually restricted to Jews and have expanded due to probably drift. When we say drift in genetics, we just mean to say luck, imposed by strong founder effect, which is very, very few individuals that have allowed to the huge expansion of the Ashkenazi community. And it's also important to say that these are not 3 statistical mothers. These are 3 real mothers that we can date by the rate of mutations of the mitochondrial DNA. And we know that they were really walking around us within the last 2,000 years. We can redo it again for all the Jewish communities. For Ashkenazi Jews, who are estimated at 8 million people, 4 lineages will explain 40%. For Azerbaijani and Georgian Jews, you can see that one lineage, different lineages, will be almost 60% of their maternal ancestry. And we go down, and we see that it is not always a founder effect. For example, if we are going to look to Turkish Jews, then you can see that 4 lineages are only coming up to 17.1%. we don't have enough time to go through each of the population differently, but that allowed us to actually see the connection between the different population within the different Jewish communities. And we can see that here we see the same phylogeny, now colored by the Jewish ancestry. And we see that basically different communities, as a rule, have different founding maternal lineages, aside from Ashkenazi and Sephardic Jews, which I showed you before. And you can also see here an example of the Libyan and Tunisians, and here Iraqi and Iranian Jews. So that's how we take a look at this the mitochondrial DNA ancestry. And I will end up by thanking my good colleagues that have helped doing this from basically many, many locations. And if there are any questions, when it is allowed to ask, please feel free to reach out. Thank you very much.
Thank you. Thank you very much, Dr. Behar. I'd like to now hand over to Professor Poulton, who will really go into some of the diseases and what we can do going forward to prevent these disorders having people affected with it. So, Professor Poulton, over to you.
I'm just fiddling around with the technology as one does at my age. Here we go. Share screen. Here we go. And that wasn't what I want. And I need to go on to. Yes. Can you see that? Is that all right?
Yes. Yeah, but your camera's still off.
My camera's still
off.
Oh, okay. Right. So yes, I just I need to switch that on. How do I do that? I can't see how to do that. Sorry, just give me a second while I faff around. Sorry. Just a minute. That's it. Yes. Yes.
Yep, brilliant.
Fantastic. Sorry about that. Yeah. Here we go. And now I've managed to shrink my thing altogether. Okay, fantastic. Great. So let's go back to the beginning. Great. Now, does that come up? Hasn't come up yet.
We can see the last slide.
Yes, well, I'm not a bit puzzled by that because the first slide is up now. Well, isn't that strange? Sorry. The trouble with being old is that technology, I can't understand why that is, because I've got the first screen up on my first one up. Let me just try again. Perhaps I should just try. Maybe I should just try closing it and starting again, should I? Let's just show.
Yeah, probably.
Okay, so I think I've closed my PowerPoint. Maybe I haven't. And let's try again. Back to the beginning again. Sorry. It's nothing like having a dress rehearsal before you start. Hooray! We've got it there. Fantastic. Yes, can you see that? Brilliant. Okay. Well, thanks so much for inviting me. And thanks very much for the talk. Doron, my very first publication in genetics was about the mitochondrial Eve hypothesis. And I ended with, it was in New Scientist, really reputable journal, haha. And I ended by asking whether or not the Y chromosome would follow the same out of Africa trajectory that mitochondrial DNA had shown. So when it did, I was rather excited. I suppose I should now ask you whether or not the Y chromosome follows the same trajectory for the Ashkenazis. But I think you can answer that question at the very end, because I'm going to start on the topic. So mitochondrial diseases, diagnoses are quite rare, and Alpers syndrome, which is one of the most serious disorders that we see, is present in one in 100,000 individuals. And if you go to your doctor with a mitochondrial disease, your GP will not know anything about them. It's almost always, they'll think it's incredibly rare. But actually, mitochondrial diseases are not actually that rare. And 1 in 300 of us actually carry a mitochondrial DNA mutation. So when you're in a crowd, there's probably somebody there with mitochondrial DNA mutation. But most of those individuals won't ever have any severe symptoms. They'll probably just get a bit deaf when they get to my sort of age. But almost all of them will actually have that deafness without perhaps realizing, unless they happen to have children and have some severely affected individuals in the family. There are many types of mitochondrial disease. You may remember Charlie Gard on the news a few years ago. He was a child who was born with severe, actually recessive mitochondrial disease. I went to Great Ormond Street and his parents wished to take him to the United States for some, hoping for a miracle cure, which of course, unfortunately, never happened and he passed away. But he obviously came to medical attention in infancy. This chap wasn't diagnosed as having a mitochondrial disease until he had valve replacement, heart valve replacement, in his late 60s. So they are very, very variable. Mitochondrial diseases can affect many different parts of the body. I've got a mitochondrion drawn here in the middle. It's this sausage-shaped organelle, part of a cell. Mitochondrial diseases can affect muscle, heart, brain, the eye, liver, kidney, pancreas. Diabetes is very common, and so on. Deafness, as I have already mentioned. So they can be very different. Certainly Charlie Gard was very different from that elderly chap I showed you the picture of. So coming back to mitochondria, they're small parts of cells that are needed for making energy. And here we've got one drawn out of the thing, and they look like little sausages when you find them in textbooks, but actually they're actually a dynamic network of let's see if I can get this to work. Can I get it to work? Oh, it's not going to move. Ah, how depressing. No, maybe no. I'm sorry. Well, when I showed this earlier in the day, it worked beautifully, and if we had longer, I would go for it. Essentially, what this shows is mitochondria moving around the cell, going to the part of the cell where they're needed to make energy. And I'm very sorry that I can't, for some reason, persuade it to. Oh, perhaps I can. Hang on. Oh, hang on. That's it. Here we are. Let's see if I can get that to go. Well, and now I need to switch it to a different. Now, can you see? There you are. Can you see that? Can you see that? Great. So you can see the mitochondria in red are moving around the cell. This is in a time-lapse picture, and the green dots are autophagosomes. So this is part of the garbage disposal system for the cell. But essentially, the message is there that the mitochondria are not little sausages sitting there in isolation. They're joining this reticulum and splitting off and moving around. They're very dynamic. Okay, so I'm now going to go back, see if I can get restarted again. Now, where are we? Let's go to that. Okay, and again. All right. So mitochondria are very complex. They don't just make energy. They also have a very large number of chemical reactions going on inside. And the DNA, the blueprint, DNA, of course, as you know, is the blueprint for how everything in the cell is made. The blueprint for this is there are two sets of instructions. There's nuclear DNA, here in the cell nucleus, and the mitochondrial DNA, which is present in all of the mitochondria around the cells. And it's sort of kind of like when you get your new camera, you have now you have a CD which contains a vast number of complicated instructions. That's like the information in the nucleus, and you also get a card which gives you the information you need to get started. So in order for mitochondria to work, you need both sets of instructions. And the kind of diseases we're talking about today are ones that actually involve the mitochondrial DNA itself, not the nucleus. I mean, the nuclear genes can cause mitochondrial DNA problems in that you can inherit nuclear gene problems that give you inaccurate copying of your mitochondrial DNA, but those will not be maternally inherited, which is the key for the mitochondrial diseases we're talking about today. So, as I said, mitochondria are maternally inherited, and the reason for that is that the sperm, here drawn in this picture here, is like a tadpole. It's really just a nucleus and a tail. It's hardly got any of the cytoplasm, the cell contents containing mitochondria, just a few mitochondria around its little neck there. And whereas the egg is packed with mitochondria, and there are about 100,000 mitochondrial DNAs in every egg. And you can see how the sperm here is about to penetrate in the egg. And what happens is that these mitochondria are shed from the sperm before it actually enters the egg. As it burrows in, the mitochondria drop off and are destroyed. And so all of the mitochondria in the single cell embryo, the zygote, come from the egg. So inevitably, diseases of mitochondria are going to be maternally inherited. They really can't be paternally inherited. So when you come to thinking about diseases, is it mitochondrial or nuclear DNA? Well, it's almost always going to be nuclear DNA. Mitochondrial DNAs are very much diseases are in the minority when it comes to severe diseases. And you'll be, of course, all familiar with autosomal recessive inheritance, which is Gideon's bread and butter, where you've got each mother and a father being carriers and ending up with a 1 in 4 transmission to children. So that's not what we're talking about today. We're talking about the mitochondrial DNA, as has already been explained by Doron. So this was one of the first patients I met where I started to thinking about mitochondrial inheritance. So you can see she's a patient who was diagnosed as having mitochondrial disease when she was about 13, because at that stage she was getting weak. She couldn't walk very well because she got what's called the spastic paraparesis. So she got stiff limbs, and you can see how she's wearing these splints on her ankles to try and keep her feet in a sensible position. And she developed poor vision. So she had a retinal degeneration. She was intellectually delayed. Her brain was rather shrunken. She developed a cardiomyopathy, so her heart wasn't pumping very well, so she got very fatigued. She developed diabetes, and she passed away when she was 31. So when we look at her pedigree, her family history here, this is her here, it turns out that her mother developed problems in her 40s, quite similar, movement problems, difficulty getting about, and then died following what seemed to be a stroke, but we would now call it a stroke-like episode for mitochondrial disease in her 50s. Her mother, before her, had also had poor coordination, which was not actually diagnosed as mitochondrial disease, but I'm sure that's what it was. As far as the rest of the family went, there was a baby who died aged 2 days. Nobody ever found out what killed him. But it seems very likely that it would have been mitochondrial disease. And a sister who died when she was about 10 from a sudden heart problem. So she probably had a cardiomyopathy, a heart that wasn't pumping properly for a long time, and suddenly nobody actually made a diagnosis of it, and she died suddenly. And so the sister I was talking to was this other sister, and she told me how she realized when they explained to her about mitochondrial disease running in her family that she had a risk of transmitting it to her children. She had no idea how big the risk was, but she decided that because her family was such a disaster area with this severe disease, that she wasn't going to have any children. And that had actually, for her, spoiled 2 relationships. She'd had 2 husbands, no children, and this had been a big issue for them. And so at that point, I felt it was really very important to try to understand whether there was anything we could do to help these patients, because I had, at the time when I went to visit them, I had a toddler, and I could just see a source of such happiness. What a terrible thing this was for the family. So this is a cell. This is illustrating mitochondrial transmission. So in that family, the reason that my patient was very severe and the grandmother was rather milder and didn't die young at all, was because of the dose of damaged mitochondrial DNA, because those patients had normal and damaged mitochondrial DNA, as in this cell. So this cell's got, say, 20% mutant mitochondrial DNA or damaged mitochondrial DNA, and 10% and 80% normal mitochondrial DNA. And you can see how when a cell divides, usually the mitochondria will partition equally into each half. But as you go multiple different cell divisions, things can vary. And so this cell has given rise to daughter cells of going from 10% up to 90% mutant mitochondrial DNA. And you can kind of imagine how when they've got a small amount of damaged mitochondrial DNA, they function pretty well. And above that, if they've got a large amount of damaged mitochondrial DNA, they may be severely affected. And you could see that there's actually often a threshold below which the cells are fine and above which they start to malfunction. And so many mitochondrial diseases fit this kind of a model. The higher the level of damaged mitochondrial DNA, the more likely the patient is to be affected. If you've got a low level of damaged mitochondrial DNA, you may never know. So here I've got an illustrated woman, say 30% mutant mitochondrial DNA. She's one of the ones where she's going to be she's a bit deaf when she gets to my age, but she's had children with, say, 80% damaged mitochondrial DNA, severely affected, 0%, no problems at all, and 30% just like her. How does that happen? Does it arise as the children develop from oocyte onwards, or is it actually something there originally in the eggs, in her eggs in the ovary when she ovulates them? Well, some work we did at the very early days showed us that actually it's this situation here where the seed it's in the eggs and the early embryos that the dye is set, and it's not so much a gradual change late in embryogenesis. And so the dose is apparent in the mother's eggs. And that was actually a very useful finding, because it meant that we could actually go ahead and do genetic counseling based on eggs, and also work out whether or not individual embryos had a high risk of the child developing problems. So prenatal diagnosis became possible, and also mitochondrial replacement therapy in its turn. So what are the solutions for this woman? She's got, as I said, it's she's got this problem that even though she's got quite a low level of mutant herself, she's able to generate children with very high levels of mutant who could be severely affected. Well, one option that we have explored is oocyte donation. If somebody who hasn't got a mitochondrial disease in their family donates their eggs, you can fertilize them with her partner's sperm in vitro and pop the embryo back into her uterus, and that child has got no chance of inheriting her damaged mitochondrial DNA. And so oocyte donation is actually a very good option, in that it's, it completely excludes the risk of mitochondrial DNA disease, and provided the egg is in the clear, and the mother is then the biological mother of because she's been carrying the baby and cares for it, as well as having the birth and then caring for the baby, and the father, her partner, is the father, but obviously she's not the genetic mother of her baby. And I think it's worth reflecting that 30% or so of pregnancies in this country are not planned. 1 in 10 of us don't identify our genetic fathers correctly, and actually, I know 4 children born by sperm donation very well, all of whom really bond very well with their fathers, and knowing that they're not genetic relatives. So I don't think myself that being a genetic relative of your mother is necessarily required for you to bond with her, and that this is a very good solution that we could actually explore more frequently than we do. It's actually very difficult to get donated eggs. So another option is pre-implantation genetic diagnosis, and here what happens is the mother is superovulated in an IVF clinic, and the eggs are fertilized with her partner's sperm in a dish, and then a single cell is taken from the embryo to see whether it's got a high mutant load or a low mutant load or an intermediate one, in order to try to work out what the risk to the baby would be. And this is a picture of pre-implantation genetic diagnosis. You've got one of those embryos with it here being held by a holding pipette here, and the sampling pipette has pushed through the membrane around the embryo and pulled out this one single cell, which will then be analysed for how much damaged mitochondrial DNA there is in it. And this is a very useful way of doing things, but it's it depends on the mother obviously having enough eggs with a reasonably low mutant load in order to end up with an embryo that which is worth putting back in the uterus. So I'm now going to get onto the 3-parent babies option of nuclear mitochondrial replacement therapy, or nuclear transfer into donated oocytes. And so what happens here is the mother is superovulated. Here I had her oocytes with damaged mitochondrial DNA in them, and the nucleus is taken out, and donor oocytes with blue, no, so without mitochondrial DNA disease, or embryos at the same stage are taken out, and the nucleus is taken out of those, and the nucleus here is put back into either into the egg or to the embryo. So essentially, it's a question of the nucleus of the being taken out at the same stage of in the two things, the donor or the recipient. I'll just show you that on the next stage. And of course, you've got 3 parents for here, because you've got the donated mitochondria, you've got the mother's nucleus, and you've got the partner's sperm. And of course, you've also got the same in the oocyte donation, or well, not the same, but equivalent. So we'll just run through it again. So what we've got here is we've got the mother's damaged mitochondria containing and the nucleus that's arisen as a result of fertilization with her partner's sperm. And here you've got the donor oocyte from which the nucleus has been removed, and then you have the nucleus is then put into with the cleaned up clean mitochondria, and so you've got the 3-parent baby here. And when this was first done on monkeys, these are the 2 first monkeys that were born by this way, from Mito and Tracker, and the 2 groups who've been pushing this are Doug Turnbull in Newcastle and Shukrat Talipov in Oregon, and they actually do it at slightly different stages, which is why I was stuttering slightly when I was talking about them being at comparable stages. But essentially, it's the same effect. You've got 3 parents. You've got the father's sperm, the mother's nucleus, egg nucleus, and you've got the recipient mitochondria, all there together. So, as I say, these are the options available, and 3 parents in that, and 3 parents of the oocyte donation, because you've got the mother being the biological mother, her partner's sperm, and the donated egg. So each of these is a 3-parent baby. So it's like changing a laptop battery. Well, I mean, that's what they were saying in the publicity about it. It seems simple, but of course, actually, it isn't particularly simple. And there was a lot of hype at the time, 2010, when this was first discussed, and we were told that it could be made available to women within the next 3 years. But actually, we still don't yet know about the results of this. So it's exciting basic science, and it's not really very relevant for most families, even with severe mitochondrial diseases. Alternatives available for most mitochondrial inherited diseases, and all really because of the oocyte donation. And I thought I would tell you about a family who I knew, who came to my clinic, where essentially the mother came with a severely affected child with 80% mutant of one of these nasty mitochondrial DNA disorders. And we checked the mum to see how much mutant she was carrying. Actually, she'd got none. We couldn't find it in any part of her. And so I said to her, well, look, I think you've got a pretty good chance, if you got pregnant naturally, of having an unaffected child. But she'd actually been following the mitochondrial replacement therapy stuff on TV, and been actually working as an advocate for it on television, and yet it wasn't yet available for her at that stage. So she asked if she could go for a pre-implantation genetic diagnosis. So we superovulated her and collected the oocytes, and actually none of them had any damaged mitochondrial DNA in them. And so they sampled one, collected one, and popped it back in her uterus. But unfortunately, the pregnancy failed, because it's actually only about 40% of each cycle is likely to be successful. And so she then got pregnant naturally, and we sampled with some CVS. So we sampled a little bit of placenta early in the pregnancy and found there was a 0% level of mutant, and so the baby was born. And you can see here, got a happy result, a nice normal baby with the handicapped child. So that was a very happy story. But we still have, as I say, so here are the options. You can either get pregnant naturally, and then you run a high risk of having affected children if you've got a high level of mutant. You can try prenatal diagnosis with a few of these disorders. I mean, they actually vary quite a lot. There are some where having a CVS, a bit of placenta sampled, works very well, but most of them don't, because the problem is if you get an intermediate level of mutant mitochondrial DNA, you don't know how severely affected the child is going to be. There isn't a really very close mapping between those. There is for the very high levels and the very low levels, but not for the intermediate ones. Oocyte donation, as I say, is a very good option. Preimplantation genetic diagnosis is a very good option. And mitochondrial replacement therapy is an option that is now available, but as yet we don't know what the results are. So sort of just run through the other issues. I guess there's still quite a lot of people who are opposed to this on ethical grounds, and obviously we're going to hear more about that from the rabbi. I personally, I'm actually a church-going Christian. I don't think that every sperm is sacred, and I don't think every egg is sacred, really, because, or even every embryo is sacred, because if it can become twins, then I don't really feel it's an individual entity as yet. So I think that a zygote, a single cell embryo, is too far, is a cell. It's too early in the process to consider it to be an individual. So I'm happy with the idea of doing things like pulling out the nucleus in terms of the ethics, because I don't think it's that far on. But what I do think is that safety is important, and safety is not yet established, really, because we haven't yet heard how the first few babies have fared. The truth is that if I was running the health service, I would be a very conservative person, and we do need a few crazy bull in a china shop people to make medical progress. So actually, we shouldn't just assume that everything can't be totally safe with new medical things. There may be long-term sequelae for the baby in old age. If you look at the mouse models of this kind of thing, sometimes if you've got mixtures of damaged mitochondrial DNA and if you've got mixtures of normal mitochondrial DNA in mice, that can be a problem. And what we know is that when people have done mitochondrial replacement therapy for infertility, and where the mother's mitochondrial DNA appears to be normal, she's not carrying any damaged mitochondrial DNA as far as we know, but they think that there may be low levels of damage that they can't detect. Some of those children, because it's very difficult to remove all mitochondria sticking to the mum's nucleus when you pop it in to the pristine oocyte, because of that, some of that mitochondrial DNA can increase in proportion. You can actually get children with 60% of the adherent mitochondrial DNA populating the child at birth. And that happened in about 1 in 6 of the first babies born where this was done for infertility. So if that happens, then the child, with a mitochondrial DNA pathogenic mutation, then the child would be likely to have some symptoms. So that's a bit of a worry. We don't understand it very, very, very clearly yet. So the risk of transmitting to the next generation is reduced, but it's not eliminated. Confidentiality. Well, obviously the reason we don't know very much about what's happened so far in the first few babies who've been born is that we need to protect the families, etc. But actually, my feeling is that although that's got to be balanced against the risk for the whole population, I mean, we all really need to know about the success rate before we can be referring more families on for this procedure. And I feel very strongly that it really is about time that the publication came out so that we actually know what's happening to these children. Does the mitochondrial donor have any rights over the child? And there was a big campaign to push it through the regulators. So I personally think that that there are quite a few unknowns that would have been quite nice to know about before it was taken forward. And I'd just like to say that my work on this has been helped by a very large number of people, and to thank everybody. And the Lilly Foundation, and actually the hospital charity, which I forgot to put on here, are the people who fund our work at the moment. So sorry, that's all probably a bit too fast and a bit too long, but anyway, I'm done. Shall I take my slides off, shall I?
Yes, thank you very much, Professor Poulton. So I'd finally like to introduce Rabbi Gideon Weitzman, who is the head of the English-speaking section of PUAH, where he counsels thousands of couples worldwide regarding fertility, genetics, intimacy, and other related subjects within the framework of halacha. Rabbi Weitzman received rabbinic ordination from the Israeli chief rabbinate and holds an MA in Jewish education from Yeshiva University. He has published widely in Hebrew and English in medical journals and halachic publications, as well as 4 books on the thought of Rav Kook. He has taught and lectured worldwide in yeshivot, rabbinic institutions, universities, and medical conferences. Rabbi Weitzman is also the rabbi of Merkaz Modi'in community. He is married to Rivka and have 6 children and 8 grandchildren. So, Rabbi Weitzman, over to you.
Thank you very, very much. So we've heard a lot about the medical side, and I'm not going to show slides. I'm just going to speak for a few minutes. And I'm going to take it off of what Professor Poulton, thank you very much for your lecture. And you sort of set me up in your last slide because you said the rabbi will answer this. So that's, I guess, my job. So I'm going to start with what you said about is every sperm sacred, which of course reminds us of the Monty Python skit, is every sperm sacred? And clearly in Judaism, we have a very different approach. When we're looking to save life and to have healthy children, which is exactly the spec that you spoke about so eloquently, it's clear that that takes precedence over other problematic things such as wasting sperm or wasting eggs or destroying embryos. So I think that the whole question of whether we can do an IVF, in vitro fertilization, which was first successful, as you know, in London in July 25th, 1978, when Louise Brown was born, I think that's already a moot point. It was true that almost 50 years ago, 40 so years ago, it was a debate as to whether we are able to use IVF. I think that's pretty much accepted today. What is debatable, and that's really appropriate to the subject this evening, and I thank I'm also Gideon, so I thank Gideon Schwartz as well for the opportunity to address you, is the whole question of what's called three-parent babies. Now that's really a term that's only really used in England, and I have a feeling it was only actually used by the BBC originally. I think they were the ones that coined that term. I don't think that's the professional term. And the reality is that it isn't really a three-parent baby. And the reason is the following. And it goes back to a question which is much more basic, and much more basic to Judaism, much more basic to what is the ethical question, which is how do we define motherhood? How do we define parenthood? Well, clearly the father is clearly very easy to define. The father is the person that gives sperm. There's nothing else that fathers do other than give sperm. The male counterpart to the female, the male contribution is very, very limited. It's just giving sperm, and that we know already. Because already in the Gemara and the Talmud in Masechet Nida, and the Tractate of Nida, our sages already said 2,000 years ago, there are 3 partners in the creation of a human being: mother, father, and the Lord and God, and each of them contributes something. And what father contributes is what the Talmud calls the white stuff, the white substance from which comes the bones and the white of the eye and light skin. The assumption is that all Jews have light skin, which of course we know is not true, and various other white things in the body. And so that seems to be very clear that what the husband gives, what the father gives, what the male gives is simply sperm. So to be a father of a child, all you need to do is to give sperm. Once you give them sperm, you're the father. To be a mother is a much, much more complicated issue, because what mothers give is 2 connected but somewhat distinct elements. On the one hand, as we go back to the piece of Talmud, the Talmud tells us that the mother gives the red stuff, the father gives the white stuff, mother gives the red stuff, from which comes the black of the eye, dark hair, flesh and bone, and flesh and skin. And of course, it is God who gives the soul and the countenance and the ability to speak and think and talk, etc. So it seems to be that the mother is the genetic mother, that in order to be a mother, you need to give genetics, the red stuff, which is sort of in the addition to the white stuff that the father gives. Now, we know today, of course, that genetically the father doesn't give even though sperm comes out in a white substance, it is not the white stuff. It isn't that we divide male contribution to the white of the eye and female contribution to the black of the eye, but it is significant that this Talmud already speaks about a contribution that's on a genetic level of both parents, of both mother and father. Why that's significant is because that was a very unknown quality until very, very recently. In 1694, Hartskauer invented the microscope. And in the microscope that he invented, he looked under the microscope and saw a human sperm cell. And we all remember the picture, and if you don't, you'll Google it when the lecture's over. And it's a Hartskauer, 1694. There's a picture of a sperm cell, and inside the sperm cell is a little human being. Has all the elements you need to be a human being. And that was a belief that was extremely accepted in science until very recently. And in fact, the reason is because there was an idea that all the genetic material comes from the father. There's no genetic material from the mother. And in fact, it's even borne out by linguistics. In Hebrew, we have a word for the infertile female, akara, and infertile male, akar. It already appears in the Torah, the word akar, the infertile male. There is no similar word in English. In English, we have a woman who's not able to have children. It's called barren, at least in classic English. There's no barren man. There's no similar form of the male. There's no similar form because there wasn't a concept that there's a male that's infertile. The only infertile. 1 second, I hear that someone's drilling next door. We made it make some noise. Let's see if I can get that. Hold on 1 second.
We can't hear anything.
Ah, that's wonderful. So my microphone is much better than I thought, which is already good. So the infertile male was not a concept. Why? Men gave all the genetic material. It's only very, very recently that we started discussing genetics, and that was discussed before in one of the previous lectures, that we understand that men and women both give genetic material. And therefore, that would seem to therefore be the conclusion is that to be the mother, you have to give genetic material. There is a male genetic material and female genetic material. And so maybe the mother is the genetic mother. However, there's another element of being a mother. The element is carrying the baby, what we call gestation and birth, delivering the baby. And so you can be the genetic mother and not the birth mother. You can be the birth mother and not the genetic mother. So if we have a case in which was mentioned in Professor Poulton's talk, which we have a series of possible solutions, egg donation, or the opposite, being a gestational carrier, what we used to call a surrogate, determines that we make a distinction between genetics and birth. So when we're able to do that, which of them is more powerful, which of them is more informative, which of them is more substantive in order to determine who's the mother? That's very different than being the father. The father is only one element. You only give genetics. The father doesn't do anything other than give genetics. In fact, there's a beautiful verse that King David says in Psalm 27, Aviva imi azavuni v'Hashem yasfeini. My mother and father have left me, and God gathers me up. Says one of the commentators, a classic commentator, not a geneticist today. My mother and father threw in the genetic material. They had sexual relations, and they left me. They do nothing now. They do nothing that can determine life, and everything's up to God. And we know how much what goes on during gestation is something that we really have no control over, and also very little understanding of. We really don't understand. In fact, one of the most difficult elements of that in my work in looking at fertility and counseling couples fertility is recurrent miscarriage, because we don't really know what goes on in the uterus. We don't really have too much control of that. There's not too much testing we do. In fact, I was just, while waiting to start, I was talking to a young woman, or leaving a message for a young woman after consulting with one of the top doctors in Israel about this whole question about what goes on inside the uterus. We don't really know that as much as we know genetics, as we understand what's going on in the Petri dish. And that came clear because we're talking about pre-implantation genetic diagnosis, PGD, in which we're able to really come to determine which embryo is carrying this genetic abnormality and which not. We can't really do that in the uterus. But which of them is more powerful? Which of them is more determining who is the mother? Is it genetics, or is it birth? And the reality, if we look at it in an ethical sense, we can't really determine it. Also, biologically, we can't determine it, because without genetics, none of us would be here. But without our mothers carrying us and delivering us, we also wouldn't be here. So which of them is more important? We don't know. It's very fascinating, at least to me, and I hope to you as well, is that our sages did speak about this. They did speak about genetics, and they did speak about birth, and they discussed this idea. And we'll give just a little bit of an example. We said it before. The most classic Talmudic passage speaks about the parents are the people that give genetic material, which seem to suggest that the mother is a genetic mother. However, there are many sources that seem to suggest opposite. There are sources that speak about we're taking the most simple thing. How do we determine who's Jewish? Is by having a Jewish mother. How do we know have a Jewish mother? The Talmud says in Masechet Yevamot, in the Tractate of Yevamot, that if we have a woman who gives birth to those children, then we determine that they're Jewish because she gave birth to them. They're not because she gave the genetic material. Judaism isn't detected by genetic material. It's detected by giving birth, by being born to a Jewish mother or converting into Judaism, which is a whole different question. But we determine Judaism by who your mother was at the time of birth. In fact, it says about Queen Esther that she was an orphan. She had no mother and father. It says the Talmud in Tractate Megillah. Why does it have to say that somewhat superfluous? She was an orphan, had no mother and father. That's determining. If you don't have a mother and father, you are an orphan. That's what it means. And the Talmud says that she had no mother. When she was conceived, when her father gave the sperm, had sexual relations, he died. And when the mother was about to give birth, she died. She died during childbirth. Says Rashi, Rav Shlomo Yitzhaki, the most important commentator on the Talmud, on the side of the page, he says, the father died when he gave the sperm, before he was able to be called father. Because at the initial point of sexual contact and sexual relations and ejaculation and the sperm entering into the vaginal tract and into the fallopian tube, we don't yet have fatherhood because it haven't yet had fertilization and implantation. And then he says, and the mother died in childbirth before she was able to be called mother. That seems to suggest, Rav Shlomo Yitzhaki, writing in the 13th century, 12th century, says that motherhood is determined by the woman who gave birth. And so the reality, okay, that's very nice for a lecture. For me, it's already 20 to 11 at night here in Jerusalem. That's very nice, but what does that mean? What do we do, as we would say, lemaisa? What do we do in actual fact? And the reality is that generally the halachically accepted position today is that we have it as a doubt. It remains a safek. We don't know. And therefore, if the mother let's take the case we said before, an egg donor who from a non-Jewish donor, the child is questionably Jewish because if the genetic mother is the mother, the child is not Jewish. And that the child would need to have some sort of conversion process. The same would be the opposite. If we have a Jewish egg, Jewish couple produce an embryo in the lab and they implant it into a non-Jewish genetic gestational carrier, non-Jewish surrogate, the child also, because the birth mother may be the mother, would also need to be converted. Rabbi Willig from Rav Mordechai Willig in New York told me that he sits on the Beit Din of America, probably one of the most the widest ranging Beit Din in outside of Israel, in the religious court. And he said they had a case in which in the morning a woman came and she'd had a non-Jewish egg donor and they converted the child. And in the afternoon a woman came and she had a non-Jewish gestational carrier and they converted the child. He said, well, one of those children is Jewish. But the answer is we don't know which. And therefore, out of this doubt, we will convert both of them. And there's not any sort of conflict between those. There's no dichotomy between those. And so we don't really know. But it would seem to suggest that mostly it's birth mother. Birth mother seems to be the most formative, but there's definitely a doubt. Which brings me back to the three-parent child. So if we go back to three-parent child, who's the mother? Well, the father is clearly the person who gives the sperm, but who's the mother? Is the mother the woman who gives the egg or the woman who gives the mitochondria? Because as we know, and we just heard, that there is a small amount of DNA in the mitochondria. So therefore, if mitochondrial DNA is what determines motherhood, then the mitochondrial mother will be the mother. If what determines is the rest of the DNA, which is the large majority of the DNA, then the genetic mother will be the woman who gives the egg. And the reality is, even though we have great respect for the mitochondrial donor, the amount of DNA in mitochondrial DNA is infinitesimally small and insignificant compared to the amount of DNA that we have in the regular egg. I know that Professor Poulton now has her hand raised. Is that a hand raised? You want to disagree? You have to turn your microphone on. I can't hear you. You have to turn your mic on.
although that's what's said was said very much by the campaigners in favor of mitochondrial replacement therapy, actually there's so many mitochondria in the egg that it's actually about 30% of the DNA comes from the mother, from the mitochondrial donor.
That's also the case in which we have mitochondrial transfer, which you're having mitochondrial transplanting. It's 30% of the DNA.
So it's basically when you're doing the mitochondrial replacement therapy, the egg donor is contributing a third of the DNA to the embryo.
Okay, that has a huge impact on what we're about to say. So we generally didn't think of it as being three parents, because really the person that gives the most DNA will be the mother. Although, as Professor Poulton says, maybe we would therefore say maybe there is. I'll go back on myself and say maybe there is three parents. And then it becomes, okay, which is the most significant amount of DNA? Which has the most significant contribution? And that may be somewhat of a debate. That may be somewhat of a question. So we have two questions here. First of all, is the mother determined by the woman who gives birth, in which case where the mitochondria comes from is irrelevant? Or is it determined by genetics, in which we then have, and then do we say, is it a percentage? So 70 versus 30, is that a percentage amount of being a parent? How do we look at that? It would be interesting, and I don't know if that's even been determined, to look at it in a legal sense. If the mitochondrial donor would come and say, I want to have visitation rights for that child, I want to have 30% of the visitation rights. Would she get that or not? I don't think there's ever been a case like that. It'd be interesting if there were, and we'll see what the courts decide. Is there some sort of significance of that DNA? My feeling is probably less than we think, less than 30%. But that's just my opinion, and that's not necessarily determines what the court is going to decide. But I think that's really the question: is that let's imagine now, what if we would have if we have an egg and we have the mitochondria, one of them is Jewish and one of them is not Jewish, would the child need to be converted? Would we go back into that debate about how do we determine motherhood? These are things that are still a little bit, have not been determined. And what I've seen, and I think is fascinating, and I've said to many of my students, maybe some of the audience here would take me up on that, this would be a fascinating PhD thesis, would be to look at the change in halachic determination, into saying that in Jewish law from Louise Brown until today. And I think there's been a real turnaround in that at the beginning everybody came out against IVF, and today pretty much it's almost universally accepted throughout Jewish different denominations and different sects of Orthodox Judaism. And I think that we'll probably see that's very much part of how Jewish law in these areas, which are new areas that are not written in the classic text. It doesn't appear in the Shulchan Aruch written by Rabbi Yosef Cairo 500 years ago. These are new areas, and new areas that need to have great understanding of what's going on medically, what's going on scientifically, and a debate between the halachic experts and the medical experts is essential. That's what we do in PUAH here every day, is we have that debate, we have that understanding, we have that, and we have members of staff who are geneticists, who are ethicists, who are involved in the debate on both sides, in order to determine what's exactly going on, and then to be able to come up with a clear halachic definition, which isn't always so clear, which sometimes the clear halachic definition will be, it remains a doubt. And that's part of the beauty of the halachic process, and part of the beauty of our development. Judaism never ever rejected per se any medical advance. It never said, well, this is just out of something we can't accept. Judaism always examined the medical advances. Many of the rabbis in previous generations and in our generation are also medical experts. Some of them are also actually doctors, practicing doctors. And we have to go no further than the great example, of course, the Rambam, who was both a doctor and a philosopher and a rabbi. His mother was extremely proud of him because he had all of those good Jewish qualities that we're looking for in our children. And we always were able to assimilate the greatness of medical advances and see it really as the hand of God. In the same way that the hand of God is determined by the books that I have here on my shelf, the hand of God is also determined by the development of scientific advance and ability to heal diseases and heal ailments. And that we're able to use that. Yes.
Sorry, Rabbi Weitzman, I just wanted to Thank you so much. That was really interesting, but I just wanted to stop there because I'm aware
Very
that
good.
we've overrun, and I just wondered if there was a couple of questions from the audience, just maybe if anybody, Anthony and Joe, do you have a question?
So I've got a number of questions, but maybe just starting with Rabbi Weitzman for a moment. I mean, in a sense, the fact that you focused on the question of how would you determine whether the child was Jewish or not, whether it needed to be converted or whatever, it carried with it the strong implication, which I think is what you were saying in your closing words, that actually there's no objection at all to any of these procedures which enable a mother to then have a healthy child because of the PUAH nefesh, the overriding principle that you do whatever you can to have a healthy human being. Yeah, so that's helpful. And if I could, maybe if I could just ask Doron something as well, if as that question was very quick. Doron, are you still?
Yeah.
There you go. One of the things that you were showing them where the mitochondria, the mitochondria within the Ashkenazi population came from, where it came from within the Mizrahi population, and so on. I have heard, and is this true that you can, from that you can analyze that essentially the Ashkenazi population, the entire Ashkenazi population, came from a relatively few individuals about 1,000 years ago. Is that right? And can we do that with the Sephardi community and determine where their origin comes from?
So that's a great question, actually. And from the mitochondrial DNA per se, you can identify a very clear-cut founder effect, which is really unique worldwide. But you can't really count the number of parents or great-great-great-parents, because the maternal is first only the mother line. So we don't know nothing about the paternal line. Plus, what we see today is only the lineages that survived from the time of expansion. So the mitochondrial DNA will not be very accurately used to count, so to say, the number of founders. But the thing that you spoke about was actually done by looking to the genome level and measuring from the genome level what is the estimated number of people that gave rise to the Ashkenazi community, and it was around 300 people. So again, it should be at very early stages of population developing, but the direct evidence is not from mitochondrial DNA, it is from the whole genome.
Thank you.
I have a question. The first baby using this technique was born in 2016. How many have been born since? How many babies have actually been born using this technique?
Well, we don't know because there's no publication as yet. But for mitochondrial replacement therapy for mitochondrial DNA disease, which has only really been done in Newcastle, we know that I think last year someone got freedom of information, and it was less than 5 or up to 5, we were told. So as far as in Greece, they're doing mitochondrial replacement therapy for infertility, for maternal infertility. And I think, again, the numbers are small, probably about 10 or something like that. I don't know what the up-to-date numbers are now.
Thank you.
Gideon, could I ask a question? You're on mute.
Gideon, you're muted.
Yes, of course.
Yeah, thanks. Yes, could I ask Professor Poulton a question? You referred to the patient, I think the young lady that had the onset at around the age of 13. It only became apparent. And what I'm just interested to understand is, so my company we work on the epidemiology of genetic diseases, and one of the challenges of that is understanding the level of penetrance of these conditions and the likelihood of them being expressed. So for these mitochondrial conditions, what are the penetrance issues around those? Are there huge variances? Are they, some of them 100%, some of them you may just carry the gene and may not be expressed at all, or is it a complete mix, just using that particular example?
I mean, we have to distinguish between homoplasmic and heteroplasmic diseases. So homoplasmic diseases are where you have 100% the same mitochondrial type in all parts of all cells. Heteroplasmic ones are the ones we were discussing where you're talking about mutant load determining how severe the phenotype is.
Yeah.
In the homoplasmic ones, for instance, there's a common one called Leber's hereditary optic neuropathy. And those patients, if you're a boy, you've got a hugely increased risk of having a problem. If you're a girl, it's much lower. It's something like threefold less. No, sorry, male to female ratio is about 1 in 6 are males. So the males are 6 times commoner than females in affected individuals. And so, you know, basically with that condition, everybody in the maternal lineage will carry the mutation at 100% level. But what it is that makes them develop symptoms of blindness, losing vision in one eye, followed by the other eye over days, weeks, or months. It's an awful thing to happen, and it happens in adolescent boys, or when they go to college. Often it's when they take up smoking, cigarette smoking. So I mean, so penetrance is a real problem. I mean, there's another disorder where it's precipitated by being given aminoglycoside antibiotics. They tend to go deaf, but for the general ones, that's where I'm saying it's depending on the level of mutant load. People have tried to make algorithms for the commonest mutation 3243, and they found it very difficult. You can't really predict. It's very difficult. I mean, the very high level needs, and they're more likely to be sick, but you can't just trying to make predictions for individual people is just not.
But so then the behavioral risk factors, such as those that affect chronic disease, will have an impact on the phenotypic expression, and that, such as smoking. Then
yes, that's right. But that's only one disorder where we know that happens.
Okay, it's interesting. Thank you.
Apologies, the classic error. I just wanted to wrap up by thanking all 3 of our speakers. It's been a fantastic discussion, debate, and I'm sure if anybody wants to reach out after this through genetics, we can direct them to our speakers to answer the questions. And to thank all of you for coming. Apologies, it's run on a bit, but that's always a sign of a really interesting lecture. So hopefully nobody minded. And just to say that the next one in our scientific series will be around Inherited Cancer Awareness Month, which I believe is September. So we will let you know when the next one will be. And if there's any topics that you would particularly like us to cover, then please reach out and let us know, and we will try to accommodate. So thank you very much, and to get on for chairing tonight's event. Thank you.